Hello everyone, I'm excited to be here today to share my project under the DRI Championship Program, titled A Comprehensive Dataset Capturing Brain Signals from Individuals with Cerebral Palsy Across Diverse Populations. I'm Elham Reyhanianmotehaver, a PhD student at Queen's University, working in the Building and Designing Assistive Technologies lab supervised by Dr. Claire Davis. This project is an important step toward improving our understanding of cerebral palsy and its rehabilitation through data science and neuroengineering. Cerebral palsy or CP is the most common cause of physical disability in children, affecting nearly 3 in every 1,000 infants. This neurological condition results from brain damage during its developmental phase, leading to challenges in movement, speech, cognition, and in severe cases, seizures. While CP is not progressive, its effects are permanent, requiring long-term management. The most common causes include infections, head trauma, and in rare cases, genetic mutations. CP is categorized based on the location and severity of the brain lesion into four groups of spastic, dystonia, ataxic, and mixed, which significantly impacts movement abilities. So, there is a need for high quality CP datasets. Advancing research in CP requires high quality databases that capture biological signals from individuals with this condition. To assess the availability of such datasets, we conducted a comprehensive research in three major databases: PubMed, Embase, and Engineering Village, using keywords related to CP, stroke, Parkinson's, MS database and datasets. The search scope is from 1993 to 2025. By structuring these searches with and/or statements, we identified how many papers focused on databases for different neurological conditions. The existing CP datasets primarily include COVID-related studies, quality of life assessments, pre-post intervention effects, genomic information, and brain imaging. In addition, the CRAN data are mostly in weird populations, which stands for white, educated, industrialized, rich, democratic. However, there are many gaps in existing CP datasets. As you can see in the figure, CP-related datasets are significantly underrepresented across all three databases. Furthermore, only about 10% of these databases are openly accessible for scientific use, creating a big barrier to advancing CP research. Another key issue is that most of available data comes from children. There is very little data on adults or elderly people with CP, making it difficult to understand how CP progresses over time. Our goal in this project is to collect brain, muscle, and motion data from adults with CP, ensuring a diverse and representative data that reduces biases associated with weird populations. Now we can ask why this dataset matters. The primary goal of this dataset is to enhance scientific knowledge about CP and address the gaps in rehabilitation research. By making this dataset open access, we can empower researchers to explore the neurological and physical aspects of CP in more depth. This dataset will allow us to analyze brain activity, neuroplasticity, and motor learning patterns in individuals with CP, leading to more personalized rehabilitation strategies. In addition, engineers and developers can use this data to design better assistive technologies, improving the quality of life of adults with CP. This dataset will focus on three types of biological signals from adults participants with CP. The inclusion criteria are a formal CP diagnosis, the ability to walk without assistance, the ability to communicate pain and discomfort. Since walking is a fundamental activity often affected by CP, we are focusing on gait related tasks such as plantar and dorsiflexion of the ankle. The biological signals we will collect include motion data recording using 3D motion capture cameras, muscle activation data using EMG electrodes, brain activity data using EEG electrodes. There are some steps to complete the project. To achieve the project's objectives, we need to follow a structured approach. The first step was to develop a detailed research proposal outlining the goals and methodology. Next, we needed to secure ethical approval from Queen's University in Canada. Since CP research requires a diverse range of participants, and regarding the previous experience of our lab mates in finding participants with CP in Canada, we needed to explore international collaborations. Conducting research in another country comes with challenges such as securing funding, arranging travel, and obtaining local ethical approval. Once all approvals are in place, we will conduct pilot tests to refine our data collection process before proceeding with full scale data recording. What is our current progress and challenges? So far, we successfully completed the proposal and defended it at Mechanical Engineering Department of Queen's University. Obtaining ethical approval was a complex and time-consuming process, but we received initial feedback from Queen's Ethics Committee 3 weeks after submission. However, we paused resubmission to align the Canadian ethics application with the requirements of our collaborator country, Iran. Meanwhile, we established strong connections with our Iranian collaborators, ensuring access to a sufficient number of adults with CP. We also applied for additional travel grants to support the financial aspects of this research, and we hope to get these grants to cover the expenses. Currently, we are seeking ethics approval in Iran while simultaneously recruiting participants. Despite our efforts, the complexity of the process has led to delays, and we have not been successful to record the full dataset yet. Although we have not yet completed full data collection, we have conducted initial tests in collaboration with brain research labs. We have EEG signals from 21 electrodes placed over the motor cortex, EMG signals from 5 lower limb muscles, including the tibialis anterior, gastrocnemius, rectus femoris, vastus lateralis, and biceps femoris. These brain and muscle signals are synchronized, allowing us to analyze motor function in participants with CP. Additionally, 3D gait analysis was conducted, capturing gait phases in 3 individuals: a typically developed person, a person with mild CP, and a person with severe CP. That you can see in 3 rows of this figure. From this figure, you can see that crutch gait becomes more pronounced as CP severity increases. This dataset, once completed, will be made open access for further researchers. Here, you can see uh, the project timeline, the next steps. We expect to receive final ethics approval in both Canada and Iran by March 20th and complete full data collection by April 20th. Following data collection, we will analyze the results, publish research papers, and share findings with the scientific community. Many thanks for your time and attention. I'd be happy to answer any questions or receive feedbacks. If you'd like to connect further, feel free to reach out through email at elham.reyhanian@gmail.com.